welcome to New Asked Practical Science. My name is Dr Cairns and today we're going to be looking at investigating enzyme action. So our aim for today is to investigate the effect of an enzyme on a large carbohydrate such as starch. You will be working scientifically to use appropriate methods in experimental work, so that'll be me. Complete a hypothesis and prediction, that'll be you. Make and record observations, present observations in an appropriate table and evaluate data. So those last 3 points are all you. Okay, so before we start, we also need to remind ourselves of safety. You probably noticed I'm wearing my goggles, okay? We need to wear our goggles for this practical. Also, as we go through the practical, I will point out certain chemicals have certain warnings on them. I'll make that very clear as we go through the practical. Right, let's have a look at the equipment we'll need. So, the first thing on my list is eye protection. I've got that on already. The next thing we need is 2 boiling tubes. So, I have 2 boiling tubes. Then we have the boiling tube rack. That's what the blue thing is here. Next, we need a 10 centimetre cubed measuring cylinder. That's what I've got here. After that, we have a glass rod, 2 pipettes, and a spotting tile. You'll notice on the spotting tile there are 12 little wells. We'll need those to look at the reactions over time. So we also need a stopwatch, some label pens, and iodine solution. Now, my iodine solution has this warning on it. This warning means that this iodine solution is an irritant. We need to be very careful when using this. I also have a 1% starch solution. Okay, this has been made up for me, pre-made up. I'll just give it a little shake. And I've also actually decanted some into a small beaker. I also have a 1% carbohydrate. Now on your practical sheet, it says diastase. However, I have a slightly different one called cellulase. Okay, they are both carbohydrates. And the last thing that I need is some distilled water. And I have that right here. Okay, so the first thing I would like you to do is to have a look at your hypothesis on page 2. I would like you to complete your hypothesis. So pause the video now and complete that for me. Okay, so I'd like you to skip forward now to the method section. We're going to review the method before we carry out our prediction and the actual experiment. So according to the method, we need to collect spotting tile and label 1 row A and 1 row B. So that's what I'm going to do now. Okay, so I've now labelled row A and row B exactly like you would see in the diagram on your sheet on page 2. I need to add a small drop of iodine into each well using a dropping pipette. There we go. So this bottle has a dropping pipette on top. And remember, iodine is an irritant, so I need to be very careful when I do this. Okay, so I've been very careful and putting just 2 drops into each of those wells labelled A and B. Okay, what's it say next? it says collect 2 test tubes and label these A and B. I have my 2 test tubes. You can see I've labelled them A and B. So I'm prepared up to this point, but what I'd like you to do is have a think about what I'm going to say now. I want you to think about how I'm carrying out this experiment and what I'm hoping to achieve, because this is going to help you with your prediction. Okay, let's have a look at what we'll be doing in this method. What are we hoping to achieve? So the first thing I'm going to do is I'm going to think back to what I already know. And I already know, and you should already know, that starch, when it's added to iodine, what colour does it turn? Have a think about that. Yeah, that's right, blue, black, well done. Okay, so it turns from this orangey colour that you can see here in these wells to a blue-black colour. Okay, so if starch turns blue-black, what do you think would happen when you add an enzyme? Now remember what enzymes do. Enzymes, in this case, in this particular enzyme, they break down a substrate. Our substrate is starch. So enzyme, this particular enzyme, which is cellulase, breaks down starch. Breaks it down into what though? Yeah, that's right, simple sugars, such as glucose. Right, so now that we know that starch turns blue-black with iodine, we also know that enzymes break down substrates such as starch into simple sugars. Okay, so we've got those points. So what do you think would happen when I add my starch to my cellulase and I watch over time? What do you think the colour change is going to be at the beginning compared to maybe a couple of minutes later? Well, that's what we're going to have a look at. Pause the video now and make your prediction. Right, have a look at the method section. We're going to carry this method out step by step. So first, I'm going to collect a spotting tile. Okay, I've got my spotting tile here. You can see I've already labelled one row A and another row B. Okay, I've added a small drop of iodine. I've actually added 2 drops of iodine to each well using a dropping pipette. And I've done that really carefully because we know that iodine is an irritant. And you can see, yeah, that's the dropping pipette. Okay. Then I need to collect 2 test tubes and label them A and B. So in my test tube rack, I have my 2 test tubes, A and B. Now I need to measure out 5 centimetres cubed of starch solution using the measuring cylinder. So here's my measuring cylinder. Here's my starch solution. Okay. Best way to measure this out is to use a pipette. Because what would happen if I was just to pour that into this very, very narrow top? Yeah, it would probably spill everywhere. So in order to be safe in the lab, especially when working with chemicals, we need to think logically and safely. So there we go. Now you've probably noticed that the starch has settled a bit in this solution, so I'm just going to give it a quick mix before I add it to my tube. There we go. Now we said we needed 5 centimetres cubed. Okay, so I've measured out 5 centimetres cubed in my measuring cylinder. Just going to show you that now. Okay, I'm going to pour it into test tube A. There we go. I need to measure out another 5 centimetres of starch solution using the measuring cylinder, and I need to add it to test tube B. Now for this I can use the same measuring cylinder because there will be no cross-contamination. That means no mixing of chemicals. Okay, adding that to test tube B now. Okay, I now need to measure out 5 centimetres of distilled water using a measuring cylinder and add it only to test tube A. So the next thing we need to do is measure out 5 centimetres cubed of water, so distilled water, using a measuring cylinder. I have a fresh measuring cylinder and I have my distilled water here. 5 centimetres cubed. There you go, 5 centimetres cubed. Okay. I'm going to add this to test tube A. I'm going to mix the contents immediately using a glass rod. Okay. So I'll need to wash that. Okay. Next bit. Measure out 5 centimetres cubed of 1% carbohydrates using a measuring cylinder and add this to test tube B. Okay, now mix the contents immediately after I've done that, and before we actually perform the experiment, I'm going to explain to you what's going to happen and when, because it's quite time dependent. So we know that in tube A we have got water and starch, and in tube B we're going to have starch and the cellulase. Okay, that's the enzyme. Now, as soon as I add the cellulase and give it a mix, I'll be adding a couple of drops to the spotting tile. Okay, now in my spotting tile, I already have my iodine in my first well, so I'm just going to add iodine to the other wells as well. Okay, so my iodine is ready, tube A is ready. As soon as I've added the enzyme, given it a mix, start my stop clock, I'm going to add it to my wells at every minute. Make sure you are recording the colour change every minute. Okay, so here is my cellulase, 5 centimetres cubed. It's going into tube B. Okay, I'm going to get a mix. I'm going to use a pipette, the cellulose one, give that a mix. Okay, 2 drops into B, start the timer. Okay, I'm also going to do that with A. And now we wait. Notice the colour at 0 minutes. Okay, it's turned blue-black. So what can you conclude from that? Has the starch been broken down yet? So our next time point is 1 minute. Okay, so at 1 minute, our solutions are still blue-black when mixed with iodine. Okay, coming up to minute number 2. Now you may have noticed that I've put B on the well first. It doesn't really matter if you do A or B first, as long as you know which one's which. Okay, so our solutions are still blue-black. Okay, so we're coming up to minute number 3. Two more drops. Still blue-black at 3 minutes. Okay, we're coming up to our 4th minute. Still blue-black at 4 minutes. getting ready to add our sample at 5 minutes. Okay, so this is measurement number 6. Okay guys, so as we're coming up to the last 30 seconds of the experiment, have a look at the colour changes as a whole, so across the timeline. What do you notice? What were your predictions? Have a think about that, and what are you actually observing? So I'm just preparing to add sample to spot number 8. Okay, so looking at the spotting tile, what we can see is that the colour change hasn't occurred as probably you thought it might be. It's completely blue-black from 0 to 8. Now in science, things don't always work first time, and that's why we need to repeat the experiment over and over again to make sure we are getting valid and reliable results. So in this case, we don't see a colour change. Have a think about what could have gone wrong with the experiment. What could have happened to maybe the enzyme or the starch, or maybe the temperature in this room? At the moment, we're about room temperature, which is roughly 18 to 20 degrees. Have a think about those things when you're trying to justify or rationalise your results. Once you've completed your table, I would like to look at the questions section. So that's on page 3 and 4. I would also like you to complete the extension, please. So question 1 is describe your observations for row A and then B. And because maybe you didn't see the colour change that you were probably expecting with your prediction, give me an explanation as to why you've seen these observations. Maybe something went wrong. What could have got wrong? So if you could complete those questions and the extension, go over all your work for cups and proud, and then submit that on Teams. So that is the end of our new Science Practical. My name is Dr Cairns. Hope to see you again soon. Bye.